First, let's get a few things straight. We call it corn, scientists call it maize.
When you say maize, people around the world know what you're talking about.
This is soil, not dirt. So you're really attacking the role of the root and its relationship with dirt and water and
Soil.
such. What that?
Soil.
Soil. So
Dirt,
why?
Dirt is dirty.
Dirt is dirty. We don't say dirt.
No.
And there's a lot happening in the soil below the maize. That's what this story is about. How roots feeding and supporting the above ground part of the plant interact with millions of microorganisms below. How Ed Cahoon and Center for Root and Rhizobiome Innovation colleagues from different disciplines and different institutions, including UNL, UNMC, and Doane, are tackling this from different angles.
There's really this growing interest in the microbiome, the plant microbiome, but I think that we're a little bit unique in that we bring a bigger group together to study not just the microbiome, but also the roots and how the roots and the microbiome interact to have some effect on plant biology. Roots in general are really understudied because compared to the rest of the plant, you can't see them. They're in the soil, so it's hard to look at the structure of the roots.
Just how hard? We got a lesson from students who spent a week digging up 3,000 maize plants. So, how do we do this?
So we start off, you take a little pair of shears.
Okay.
You take our shovel in order to get the whole root ball out of there.
Okay.
You break it apart, all that dirt out of there, and you want to get at those roots.
Okay, so I'm just gonna slam this like this.
Oh yeah.
When you're doing this in the summer for about 100 or 200, you get a little worn out.
Yep, then you take your shears,
Okay.
you take your vial, you would cut those roots off and you put them inside the vial.
You see all this wonderful soil that's associated with all these roots. There's millions of microbes in here. And these microbes do a lot of work for the plants too, not just for themselves. And so they can really free up that nitrogen for the plants so that they can grow much better.
A lot of kind of mystery hidden in these roots, isn't there?
Absolutely, tons of mystery. It's like the Wild West.
Then this goes back to the lab. Okay. There we go. To the lab where freezing preserves the soil samples.
Excuse me, samples?
To a cool room for overnight thawing. To the hands of folks like Michael Meyer. He adds what's basically soap.
Just shake it vigorously to shake the soil off the roots. And this is what comes off, which contains all the bacteria we want. Transfer the soil to this 96-volt tube. This will fit into a robot that extracts DNA for us. This DNA we'll send for sequencing. Once we get the DNA sequences back, we can read that essentially like a book, and it will tell us which bacteria live in there and maybe even what they do.
This is just some of the work that's happening.
12.2,
Back in the field, students measure tassels on maize plants.
7.6.
Above ground, but still root related.
So if they're not getting enough nitrogen, they're not going to produce the ears of corn. And then a lot of the times, if they don't get enough nitrogen, they're not going to pollinate.
12.
Ob Rotshaw makes models of the different regions, zones, and types of maize roots. In layman's terms, if the root is a road, you're working on the road map and trying to determine what's happening on those roads.
Exactly.
Plants grown in glass beads instead of soil lets researchers learn more about compounds produced just by the root itself.
And so we try to create these more sterile or more isolated conditions in the glass beads that where the chemicals that we're getting should only be from the root.
There's research happening in traditional greenhouses. But also here, a huge plant phenotyping platform. It's fully automated, set up for daily analysis of up to 672 plants, moving through infrared, fluorescent, visible, and hyperspectral chambers to gather a wide range of info. Weighing and watering is also automated. There aren't many, if any, places like this in the country. It's all a big, multidisciplinary group of scientists and tools, with good reasons for digging into what happens below ground. So what's the end goal of what you're hoping to learn from all of this?
So the end goal is we're hoping we can figure out if there's certain genetics with the plants that lend towards a beneficial microbial community for that plant.
So right now you have a lot of pieces, is
Yes.
basically what you're saying?
Yes, we have a lot of pieces, a lot of good work that's been done, a lot of discoveries. But in some respects, they're in different areas of the project, and now we're really focused on just bringing them together and developing high-yielding maize that can produce with lower water inputs, nitrogen and phosphorus inputs. It's great if we can do something that really benefits the farmers of the state. I do think that the innovations that we're making will have impact